Hello, everyone, and welcome to this session of the GCI Summit. My name is Dr. Paul Gavin. I'm the Chief Executive Officer of Avecho Biotechnology. Avecho is a Melbourne-based company with a novel formulation technology called TPM. Now, our background is pharmaceutical drug development for traditional non-cannabinoid medicines. In this space, we've been using our TPM to increase the solubility and bioavailability of pharmaceuticals, and then conducting formal clinical trials to demonstrate their performance in people. Today, though, I'm going to talk to you about how TPM can be used to overcome a range of problems associated with the formulation of cannabinoids, specifically their poor solubility and bioavailability. So, look, to set the scene, in the last decade, there's been a huge shift in the treatment of medicinal cannabis following the recognition of the therapeutic potential of cannabis and cannabis extracts. Now, many countries around the world are currently enacting some sort of legislation to make medicinal cannabis legal for therapeutic purposes. Additional countries have gone further and are allowing markets in both consumer and recreational areas. But I guess from the biotech side, the fact that it's been illegal for so long means that there's really been limited R&D conducted into formulation development. And so initial product offerings to patients and consumers have traditionally been oil-based vehicles. Moving forward, a range of more acceptable dosage forms are going to be required to fulfill the needs of patients, physicians, and consumers. However, the cannabinoids themselves suffer from a range of technical issues that make formulation difficult. And so cannabinoids, which are the key active components of medicinal cannabis, are oil-soluble molecules with poor solubility in water. And like all other oil-soluble molecules, they have very poor oral bioavailability. So depending on the literature that you'll read, you'll see figures reported from potentially anywhere from 3 to 10% bioavailability for the cannabinoids. So what this means is that 90% of the cannabinoids in the product that patients or consumers are consuming is metabolized or excreted and is never absorbed to give any kind of biological value. So the low bioavailability presents real challenges for developing formulations that can deliver cannabinoids to the body efficiently. And so this has become a real focus of a lot of formulation labs around the world, because if you can increase oral bioavailability, you can potentially give greater therapeutic effect because there's more cannabinoid absorbed to exert a biological influence. You can potentially shoot for new clinical indications. So try and treat indications that ordinarily would require huge doses of the cannabinoids. You can reduce the cost to patients, which is a major impediment for them, especially in Australia. And on the commercial side, you can really provide some technical and commercial differentiation. And so this is really where Avecho has spent the last year investigating how our TPM may potentially add value to the cannabinoid space. And so what is TPM? This is my single slide overview of TPM. I'll go into a bit more detail shortly, but it's the core component of our drug delivery platform. TPM itself, it's a mixture of two different forms of phosphorylated vitamin E, which we manufacture at our facility in Melbourne. Now the TPM, because it's tocopherol phosphate, has enhanced vitamin E activity compared to normal tocopherol. And quite uniquely, when you put it into a system that contains water, whether that's a formulation or the stomach, the TPM can self-assemble into tiny vesicles that encapsulate drug molecules. And this encapsulation can increase drug solubility and stability, especially for drugs that are poorly soluble in water. So you'll hear me say that a lot in the next 20 minutes. Now, an example of that is this image that you can see here. So this is one of our non-cannabinoid formulations. This is a reformulated propofol injection. Propofol itself, the drug, it's an oil, it has no solubility in water. And so the commercial product, as you can see on the right, is an opaque lipid emulsion. Now, we reformulated it, adding a little bit of our TPM, a bit of our vitamin E, and created a transparent microemulsion, which you can see on the left. And so when we use TPM to encapsulate drugs, apart from solubility and stability aspects, it also allows us to increase our absorption, both in the oral route of administration, so oral bioavailability, as well as topical dermal and transdermal absorption. Now, it's built for vitamin E, so it's very safe. And to summarize, I guess, when we reformulate drugs with TPM, it allows us to give the products improved pharmaceutical properties and performance, and allows commercial opportunities for differentiation and patent protection. So in the next few slides, I've got a bit more detail for the formulation scientists amongst the audience. So this first slide here talks about the structure of the components in TPM. So as you can see at the top, we start with vitamin E, normal alpha tocopherol. Our phosphorylation reaction creates tocopherol phosphate, which is the structure you see on the bottom left. So it's just vitamin E with a phosphate group stuck on the end. And on the right, you see the other component of TPM, which is ditocopherol phosphate, which is essentially two vitamin E molecules stuck together by a phosphate bridge. And together, this mixture is called TPM. And this mixture always contains TP and T2P at a roughly 2-to-1 ratio, which turns out to be pretty important for a number of the applications that we use it for. Now, TPM has enhanced vitamin E activity. So we actually spent the first probably 5 years of the company life characterizing TPM as a form of vitamin E. The product was actually sold in the US on market as a vitamin supplement called Ester E. And consequently, we did a lot of work characterizing its activity. And so we published a lot and proved that TPM, or tocopherol phosphates, have enhanced vitamin E activity for a range of the non-antioxidant vitamin E roles. It has anti-inflammatory activity. It provides benefits for dyslipidemia. It can improve endothelial dysfunction. It's a potential protective agent for cardioprotection. And it's very good at dermal anti-erythema. And consequently, it's sold into personal care products all around the world, specifically around reducing dermal erythema. On the safety side, it's metabolized to tocopherol, and as I mentioned earlier, has self-affirmed GRAS status for oral consumption. And so I guess why do I mention this vitamin E activity? Because it really differentiates itself from other technologies, surfactants that you might typically use to increase absorption. And the interesting thing specifically about the TPM, it's a range of these properties that it has actually appear to be complementary for some of the indications that you ordinarily might use CBD or other cannabinoids for. So it might be that TPM can increase the absorption of the cannabinoids whilst also exerting its own additional benefits. Now, the unanticipated consequence of phosphorylating tocopherol was that it would now form vesicles, self-assemble into these tiny little nanoparticles that are less than the width of a human hair. In retrospect, it's probably not that surprising because we've taken, as you can see on the top right, a structure that has a big water-hating element on one end. We put a water-liking element on the other end. And so consequently, we've made a molecule that structurally looks a little bit analogous to a phospholipid, which everyone knows makes liposomes. And so as you can see in the images on the left, this is what a TPM colloidal suspension looks like. As we increase resolution all the way up to cryo-TEM, you can see a highly ordered vesicle structure made purely out of TP and T2P. And the important thing about these vesicles is that they encapsulate and solubilize drug. And again, it works particularly well with lipid-soluble molecules. So again, the image you see on the left, it's an example of a non-cannabinoid. This is a vitamin K injection that we've been working on. Again, vitamin K, it's in soluble oil. And on the image on the left, you can see that without TPM, vitamin K just sits as a non-dispersed lipid phase on the surface of the vehicle. However, when you add TPM, it's no longer on the surface because it's been dissolved and encapsulated by the molecules and evenly distributed throughout the formulation. Now, this encapsulation experiment, just this with and without TPM, is usually what we start with because it gives us a pretty good idea if a drug's going to plug directly into our technology. And when we can prove that TPM encapsulates drug, history shows that there's a range of advantages this can bring for other dosage forms and routes of administration. And so we've shown that it will increase oral bioavailability. So with this route of administration, when you co-dissolve TPM in an oil carrier with an oil-soluble drug and they hit the gastric space, the mechanism of action that we're sort of examining at the moment is that TPM vesicularizes and emulsifies the lipid, very similarly to the way that the gastric system works anyway. And so it may be that TPM shortcuts this process or allows it to occur more efficiently for subsequent absorption. It also allows increased topical absorption. So the TPM itself absorbs quite well into the strata corneum. It can carry drugs deeper into the dermal layers, and we've done a lot of clinical trials on transdermal and dermal absorption of molecules using TPM. And on the injectable side, it allows you to replace pretty nasty surfactants, chromophore, these kind of things, with essentially vitamin E. So as you see on the image on the left, that's actually an example of injectable formulation. And so I've just got 2 slides here demonstrating data on this increased absorption. Now, this first slide is around low bioavailability of lipids. And in this case, we've got a model drug here, which is coenzyme Q10. Now, on the left, what you're looking at there is solubility and dispersibility of coenzyme Q10 in vitro gastric absorption model. So, aqueous system meant to mimic the contents of the stomach. And as you can see, CoQ alone, which is the black line, it has pretty poor solubility in the gastric space. It stays poor if you put normal vitamin E or vitamin E acetate into the system. But if you add TPM, you can see suddenly that the solubility of coenzyme Q10 in the gastric space is increased. Now, when you turn to the graph on the right, this is what happens when you give those same formulations to animals and monitor the blood concentration over time. And what you can see, again, comparing the blue line, which is the TPM CoQ formulation versus all the others, you get a significant increase in bioavailability. And this has been true for other lipid-soluble molecules that we've looked at as well. On the topical side, you get a similar kind of phenomenon. So this is, again, animal experimentation. We've kept it constant using CoQ as this example, although we've done a range of drugs and pharmaceutical products, both in animals and human clinical trials. And as you can see, when you add CoQ alone to the surface of the skin, animal models, there's very little absorption. But when you add it with TPM, a formulation with TPM vesicles, you can see a significant increase in the amount of coenzyme Q10 that's absorbed into the skin. Now, the graph on the right is the area under the curve of the plasma concentration. So the amount of drug delivered transdermally in the same experiment. And you can see, again, when formulated with TPM, there's a significant increase in the amount of transdermal coenzyme Q10 delivered. And so you can probably all see where this is going already. So we have a problem with cannabinoids. They're oil-soluble drugs. They have poor solubility. They have poor bioavailability. We have a vitamin E-based technology that allows you to increase all these things. And so, from our perspective, if TPM did for cannabinoids what it has done historically for all other lipids that we've examined, it'd provide us with an opportunity to develop, really differentiated cannabinoid formulations that perform better than the first generation products that kind of entered the market.
Yeah.